Hello, and thank you very much for joining us. My name is Clive Glover, and I am the Product Manager for Pluripotent Stem Cell Biology at Stem Cell Technologies. Stem Cell Technologies is specialized in providing tools to standardize research with many different cell types. Today, we will be focusing on AggreWell, a tool to generate large numbers of uniform cellular aggregates of defined size. I am very pleased to present Dr. Mark Ungrin from the University of Toronto. Mark is the inventor of AggreWell and developed the plate primarily in the context of making Embryoid bodies from human embryonic stem cells. Embryoid bodies are three-dimensional aggregates of embryonic stem cells, and generating embryoid bodies is usually the first step in many differentiation protocols for these cells. Let me now hand the controls over to Mark to present the webinar. Mark?
Thanks very much, Clive. So I'll start off, you can all see the title. Clive has given the introduction there. So I'll start off with a little bit of background information around the motivation that got me started working on this project and developing this technology, and then move on from there. So when I first started my postdoctoral position in Dr. Peter Zanstra's laboratory at the University of Toronto, one of the first things I was told was go out in the lab and just familiarize yourself with hESC techniques, so passage some cells, grow some cells, and make embryoid bodies. And so I did that, made the EBs using the conventional scraping approach where you scrape colonies off of 2D culture systems. And I wound up looking at something a lot like what you see here in panel A. So this is a range of hESC aggregates, and one of the first things that strikes you about this is it's very heterogeneous, both in terms of the relative sizes of the aggregates and also the shapes are quite inconsistent. And people in the audience who work with hESCs and EBs may also be looking at this and thinking that it doesn't look anything like the EBs that you prepare. And that's a third source of heterogeneity, is that because the process for making them is so difficult to quantify, it winds up being fairly idiosyncratic. And so two people between different labs or even within the same lab will often wind up generating very different preparations of embryoid bodies despite following the same protocol. And these sources of heterogeneity are quite significant in that it means that no two cells, whether within the same aggregate or in between aggregates, are likely to have the same local microenvironment. And that's significant because of the impact that the microenvironment can have on differentiation outcomes. So inset in the upper right corner here is an immunofluorescence image of some embryoid bodies that were fixed in the process of aggregating together. So these EBs, and they were stained for CDX2 transcription factor, and I won't go into great detail about what that means in terms of cell differentiation fates and so on, but the important point is the spatial distribution of that signal. So these EBs were all in the same macro environment, the media is the same, the growth factors are the same, they're all in the same well, and yet based on the microenvironment, particularly at the intersection between fusing aggregates, this CDX2 transcription factor was activated. So this is an example of where the local microenvironment, which is an uncontrolled variable, is overriding the macro environment that is controlled by the experimenter. And so this can cause obvious problems if you're attempting to direct differentiation to specific outcomes. Now, one interesting observation that I did make, though, looking at this population of mixed EB, was that a very rare subfraction on the order of a few percent wound up looking like this, down panel B, where you have this internal sphere and then an outer shell. And this really caught my attention because this degree of order isn't something that you would expect to arise randomly. It's something that implies that the ES cells are executing some kind of inherent program for self-organization. And so that was quite interesting to me. And I started following up with a bit of immunofluorescence. And it appeared that the inner ball consisted of OCT4 positive cells, while the outer shell was cells positive for endodermal markers such as FOXA2 and so on. And the two layers were separated by a laminin-containing basement membrane. So at this point, I didn't know too much about the basics of early human development, so I did some investigations in the literature, and I'll just summarize some of that here. So on the top panel shown here is the early stages in morphology starting from the fertilized egg. You get several rounds of cell division giving rise to the morula. followed by a radial round of cell division, where cells on the periphery develop into the trophectoderm, and cells in the interior become the inner cell mass. Then a cavity opens up within this structure known as the blastocele. The embryo at this point is referred to as the blastocyst. Cells on the blastocelic surface of the inner cell mass give rise to the primitive endoderm, which then migrates out along the blastocelic surface of the trophectoderm, where it differentiates into parietal endoderm, and where it remains in contact with the inner cell mass, visceral endoderm. The visceral endoderm then lays down a laminin-containing basement membrane, and the inner cell mass polarizes and epithelializes to give rise to the epiblast. And this is interesting for a couple of reasons. First of all, from a developmental biology perspective, this is a stage of development that's very difficult to access in humans for both ethical and practical reasons. And from a tissue engineering viewpoint, the epiblast represents to tissue engineering what the pluripotent stem cell represents to cell biology. And that is because the epiblast is a pluripotent tissue. It undergoes gastrulation and gives rise to the 3 canonical germ layers, and then goes on to give rise to all the tissues and organs in the developing organism. Now, the lower panel here is simply the same differentiation process, but diagrammed rather than in terms of physical location, in terms of a differentiation hierarchy. So I then was interested in trying to replicate some of these organized structures. And as I mentioned, I had initially generated these using conventional scraping techniques to remove ES colonies from the culture surface. So these techniques generally involve either cutting up the colony to break it into large clumps, or incomplete enzymatic digestion, whether disbase or collagenase and so on, and a certain degree of trituration to break up and give a desired clump size. And as you can imagine, this is, as I mentioned, a very difficult process to describe completely, difficult to quantitate, and therefore difficult to repeat exactly, whether within a lab or between labs. So the result is heterogeneity of EB sizes within an experiment, heterogeneity of differentiation outcomes as well, and then inconsistency between experiments performed by the same experimenter and between experiments in between labs when you try to carry a protocol back and forth. So in order to get around this, I looked at forced aggregation protocols. So these are protocols where you start with a single cell suspension of, in this case, ES cells, pellet them either centrifugally or just by allowing them to settle, allow them to incubate, and they will form one, or depending on your geometry and the number of cells present, sometimes more coherent aggregates, which can then be extracted. Force aggregation is a process that's been widely used in the literature. The most common is, or most commonly known, is the hanging drop technique, which is very common in the mouse ES field, discussed along with a few other techniques in this Stang et al. 2002 reference. Mouse ES aggregation in a microcentrifuge tube was published in 2003, and then 2005 adapted for use in a 96-well plate format. And later that same year, a similar protocol with the addition of a centrifugation step was published for the aggregation of human embryonic stem cells, again in a 96-well plate format. And one point that I and others I've talked to about this have encountered is it turns out to be somewhat technically challenging to perform this forced aggregation reproducibly with human ES cells. I was fairly lucky when I first started the process. The first couple of times I did the experiment, aggregate formation occurred successfully. And then we went away over the Christmas break, and when we came back in January, nothing was working. So I spent quite a bit of time, first of all, trying to recapitulate my own earlier results, and then trying to determine why it was that the protocol that had previously been working had stopped doing so. And I won't go into great detail about that, except just to show kind of a key piece of information here. EB formation after force aggregation is strongly affected by the input population. So I'll draw your attention to panel A for a start. On the left-hand side here is a histogram from a flow cytometry experiment for the pluripotency marker OCT4. In red is the baseline ES cell population, and in blue is the profile derived from the original population, but differentiated for 72 hours prior to harvest in the presence of serum. So you can see the OCT4 peak is developing the shoulder on the left-hand side, indicative of at least a partial differentiation of the population. On the right-hand side is a bar chart showing cross-sectional area of aggregates formed from the same number of input cells either from the original ES population in red or in blue from this pre-differentiated population. And you can see there's a substantial and significant increase in aggregate cross-sectional area, which is effectively a proxy for the size of coherent aggregate that can be recovered, again, from a consistent number of input cells. Similar results in panel B here from ES cells. Now in this case, where the upper panel, the cells were grown on MEF feeder cells. In the lower panel, grown on Matrigel in defined medium. Again, in red is the baseline population, and in blue is after a pre-differentiation. And then on the right-hand side, this bar chart shows in the absence of pre-differentiation, now off of Matrigel and without any survival factors added, stable aggregates were not formed from this population. Addition of pre-differentiation did give formation of a stable aggregate population. And then addition of the ROCK inhibitor, so this is based on the results of Watanabe et al., the Nature Biotech paper showing that the ROCK inhibitor promotes survival of ES cells in single-cell suspension, with or without pre-differentiation, gave even significantly larger and stable aggregates. So this explains, among other things, the reproducibility problems I had initially, that prior to the Christmas break, we'd had late passage ES cells that were likely somewhat adapted to culture conditions and so more able to survive as single cells. Whereas after the Christmas break, we had discarded those cultures and started again with early passage ES cells, which were now not yet adapted and not prone to survival as single cells. And it also explains some of the difficulty that maybe others have encountered in this area, in that often when we encounter a problem in ES cell work, the instinct is to go for the best ES cell population we can find, which is generally defined as one that's uniformly OCT4 positive, a very high percentage of the overall population, which just so happens to be the population least likely to successfully form aggregates under these conditions. So having established conditions under which we could reliably form aggregates from single-cell ES suspensions, I then looked at ways to really increase the throughput dramatically. And after looking at a number of different potential approaches, I wound up developing this microwell technology that has become the AggreWell. So just some graphics here to show you visually how the system works. On the right-hand side here at the top, this array of squares is a top view looking down on an array of 400 micron microwells. You can sort of see diagonal lines leading from the corners into the center. These are the sides of these square pyramidal microwells. So there's 4 triangular faces that meet at a point. At the middle here is a cross-section at the same magnification of these same microwells. So you can see that pyramidal shape in cross-section. And then at the bottom, a single microwell at a higher magnification, showing that the features are actually quite sharp in this. And this has a consequence that at the scale of your typical ES cell, so 5 to 10 microns in diameter, the microwells are effectively perfectly sharp, meaning that even at tens of ES cells or any very small number of ES cells, you can put them in and they will still all slide down and encounter one another at the point. There's no surface area at the bottom for them to be scattered across. On the left-hand side is a graphic showing an image taken from the top down of a number of ES cells aggregated in one of these microwells, projected onto a 3-dimensional reconstruction from the original design. So that's a theoretical reconstruction of the 3-dimensional shape of the microwells, just to give you an idea of the geometry as it would be seen from an angle if you could get down really close. So the AggreWell 400s as currently available from StemCell are shown here. It's a 24-well plate with 8 inserts in these central wells. The microwells are 400 microns across, and the size range is down to a few tens of cells. You might even be able to go lower, although I haven't tested it quite that far down, but certainly 30 cells, and maybe I think I've tested even down as far as 15. Up to between 2,000 and 4,000 cells per aggregate is the upper limit, depending specifically on the size of the particular cells that you happen to be working with. So the overall process flow looks like this. You start off with your cell population and generate a single cell suspension. You add these cells to the AggreWell plate and centrifuge them down under the conditions you would normally use for pelleting of these cells. And as they move down towards the bottom surface of the well, they encounter these microwells. And as each microwell has the same surface area, assuming that your single cell suspension is well mixed and homogeneous, each microwell should capture approximately the same number of input cells. So the plate is then incubated for the desired period of time overnight, or depending on your cell types, could be longer. And the following day, harvested, generally by pipetting. The aggregates can then be passed over a filter. In this case, unincorporated single cells will flow through the filter, whereas the aggregates will be captured. You can then invert the filter and wash the EBs off into the plate, and you can see this sort of uniform size that we're getting in this case. Aggregate size control is a function purely of the input cell number. So, for example, in these current AggreWell 400 plate size, there are approximately 1,200 microwells within a single well. So if you want to generate EB of a given size, you simply multiply the number of cells you want per aggregate by the number of microwells. So in this case, if you want to generate 500 cell aggregates, you multiply that by 1,200, and you need to add 600,000 cells per well. 1,000 cells, twice as many input cells. 2,000 cells per aggregates, twice as many again. The histogram here shows cross-sectional area of aggregates generated from 500 cells or 2,000 cells in the AggreWell system, or in red, histogram showing formation of EBs using the traditional scraping protocol. And then on the right-hand side, photomicrographs of the wells after input of, again, 500 or 2,000 ES cells. Now, an important point to bring up at this point is that this enhancement in control over embryoid body size also increases the importance of knowing where to aim. So when you work with conventional scraping protocols, it's very much a shotgun approach. You may not know exactly where the target is, but odds are pretty good by firing at random. Somewhere within that population, you'll have the optimal size and symmetry for the cell type that you're interested in producing. By contrast, if you work with AggreWell-derived consistent embryoid bodies, one of the things is that you can start out with conditions that may not be optimized for your specific cell type, and so you can actually wind up with efficient production of something else entirely and completely miss what you're aiming for. However, once you've optimized the desired conditions, you can then hit that target repeatedly and obviously with much higher efficiency than the heterogeneous shotgun approach. So now just some data from our original paper on this that came out earlier this year. Forced aggregate human EBs differentiate effectively. So here's cardiomyocyte differentiation, hematopoietic differentiation, neural differentiation, and then these two panels here show QRT-PCR data showing upregulation of markers from all three germ layers. And back to the original motivation for developing this technology, forced aggregate human EBs self-organize efficiently. So this again is that structure that I was showing you before, but now generated using the forced aggregation approach. You have these aggregates, even on phase contrast, you can see very clear organization into a monolayer of OCT4 positive cells. Nuclei are located basally over a laminin-containing basement membrane. And cells in the outer layer are positive for primitive endoderm markers such as GATA6, GATA4, AFP, FOXA2, and SOX7. And just to kind of bring the point home a little bit, comparing that to this model of early human development that I showed in the introductory slide, you can see again we're looking at columnar morphology overlying, not stained in this particular photomicrograph, but the laminin-containing basement membrane is here, and then markers of visceral endoderm here. So I would argue that this is a model of the epiblast stage of human development, which is something obviously of significance both from a developmental biology and tissue engineering viewpoint. So just to kind of step back and take a higher level overview of the whole process, we have generation of aggregates from a single cell suspension here in theory and here in practice. You can see the numbers are quite high here. You can then allow these aggregates either to differentiate on their own using endogenous signals, where they self-organize into this structure of interest to developmental biologists, or by adding exogenous factors, you can override the endogenous signals and drive differentiation in a desired direction. Here we just have some flow cytometry data from a project where we're optimizing, among other things, EB size for differentiation to definitive endoderm, and you can see we can consistently get over 90% purity in this differentiation process. There are several papers out there in the literature about the effects of ES cell cluster size on differentiation. The Ng et al. Paper from the Elefanti lab in 2005, which was the first use of forced aggregation in human ES cells, also looked at the effects of EB size on hematopoietic differentiation. There's a publication looking at effects of ES cell cluster size on endoderm marker expression. A couple of papers from the Zanstra lab, Rahim Pirani and Celine Bowens, looking at endoderm and neural differentiation versus colony size, and also looking to some extent into the mechanisms that may be driving this. Burridge et al. Paper looking at cardiomyocyte differentiation from EBs when compared to specific size ranges. And then another paper from Elizabeth Ng in the Elefanti lab looking at neural rosette formation in larger size EBs. I've also looked beyond human ES cells, which are my primary focus, at other cell types using this force aggregation approach in the microwells. Works quite well for mouse ES cells, which have several advantages, not least of which is the fact that you can get a lot of different reporter lines in the mouse ES cells. So this is just an image of a mouse EB with GFP driven by the brachyury promoter coming up. I've also formed aggregates from mouse embryonic fibroblasts from HeLa cells. So this is a proxy for the tumor spheroid model, which is currently gaining in popularity in the area of cancer biology, both from the basic biology perspective and also in terms of studying chemotherapeutic agents, which are now being realized to behave significantly differently in these three-dimensional models than they do in 2D culture of the same cell lines. Primitive endoderm aggregates of various different sizes, and also mixing experiments. So this is an example of something that would have been very difficult to do with the conventional scraped EB protocols. Human ES cells aggregated either on their own or in a one-to-one mixture with primitive endodermal cells generated using Martin Pera's BMP differentiation protocol. So key points to take away from this now are that the EB produced are highly consistent. They can self-organize into highly ordered structures and differentiate into all three germ layers. And this provides a good starting point for investigating in vitro differentiation and all this self-organization and higher level structure that's of interest from a developmental biology perspective. It's also extremely scalable and high throughput, so you can take the results from a very small number of wells and expand very rapidly to much larger scales. I've made as few as perhaps tens or hundreds of aggregates within a single experiment, up to 900,000, slightly over 900,000 is the largest single experiment I've done. And that was limited really only by the number of input ES cells available. It was really only a matter of a few hours in an afternoon to put that together. And then, of course, it's applicable to various other cell types, including, for instance, tumor spheroids, the cell types that I've shown here. And we have collaborators who've tried it with a number of other cell types, and it seems to be universally effective as far as I'm aware to date. So I'll just end there with acknowledgments. The differentiation analysis was done in collaboration with Chirag Joshi, Celine Bowens, and Andronika. Advice and suggestions on the technology development from my principal investigator, Peter Zanstra, as well as Maxime Poirier, Aaron Wheeler, and Rahim Pirani in the lab. Human ES cell culture, the input cells that I used to generate these data were grown by Manuel Alvarez and Ting Yin in our lab. And as well, thank you to the Zanstra, Van der Kooi, and O'Day labs for general help and suggestions during this process. If anyone's interested in more details on some of the experimental work that was shown here, here's the reference for a paper that came out earlier this year in PLOS 1. That's an open access journal, so everyone should be able to download that paper. It's listed in PubMed if you need to find it after the presentation's over. If you come up with additional questions or ideas or product suggestions involving this or requests, you can check the stem cell website and contact Clive Glover, who, as he mentioned, is the product manager for pluripotent stem cell biology for Stem Cell Technologies.